Oh, there's an actual face. Hello, Jesse. How are you?
I'm good. How are you?
I'm okay. I mean, not that these pictures aren't actual faces. It's just, they're actual animated faces. This isn't what any of us signed up for. So, if I were you, I'm not sure I'd ever stop being pissed. And I think that's probably a good thing. I want you to be angry for a long time about this because it'll happen again if we're not careful. Sorry, I started my day actually reading the newspaper and then the BBC, and I'm afraid it's put me in a mood. Let me, thank you for, I'm going to push record here. If I can find it, let's see, more. Record, there it is. Okay. Paying building fees, yeah, even though almost the classes are online. Yeah, well, if of course the university is bleeding money in a lot of other ways, so you're subsidizing at this point the athletic department, but no, I don't know that for a fact, but it does feel weird that you're paying building fees. For those of you, by the way, who are going to sign up, when is registration? It must be coming up soon. Those of you who are thinking of or need to take for a lab class, the physiology labs 442, Z442, that will be an in-person class. It will have to be a bit restructured, but you will be in quarterly for that. And I will be in there for some of the labs, and a grad student will be in there for some of the labs. But it will be an in-person affair of one sort or another. But yeah, the lab's in person. So I'm trying to rectify that. I will again be the only person in integrative biology teaching in person, and tiny fraction. But I feel like for you seniors, especially, not having some experience like that is just, well, going to leave you with a bad taste in your mouth, for sure. So we'll do what we can. We'll be restricted on the number of people we can have in lab. We'll have to cycle people around, and it'll be not a complete experience that it might have otherwise been, but it'll be fun. I think you'll learn something good. And, as these kinds of situations tend to do, they tend to burn away irrelevancies and leaving only the stuff that really, really is important. And I think the lab experiences are really important. So anyway, we'll distill it down to something fun and meaningful. And I think there are 4 lab sections. That is, by the way, the absolute maximum that we'll be able to do with 16 in each lab section. And so it will be restricted to seniors, graduating seniors. If we have room after that, then other people will be allowed to enroll, but I won't be able to budge on the numbers. It's going to be what it is. Anyway, I think when we left off, it was somewhere in here. We're thinking about neurons, thinking about them in terms of the simple physics of electrical transmission. In this case, we're again not talking about electrons. We're talking about, well, I guess second or third hand you are, but we're talking about actually moving cations here. So in particular, sodium ions and potassium ions. So we were thinking about how the resistance to transmission along the long axis is going to be inversely proportional to the diameter. That is, the bigger the axon, the easier it will be for things to flow. Makes sense? What don't you like about that, computer? Okay, we'll try this. There we go. And to review, this is what's going on. We've exceeded the threshold, the trigger zone. We have electrically gated sodium channels opening. That change in memory potential causes more electrically gated sodium channels. That's not exempli gratia. That's EG is in electrically gated sodium channels open, and so it's a self-propagating thing. They call that the Hodgkin cycle, by the way, and more and more textbooks are leaving out names like that, which annoys me somewhat. It's there's some kind of politics involved here that we're trying to essentially eliminate individuals from the historical record. After all, they were just people of privilege doing work. Well, it was work. I don't care where they came from. It was work, a lot of it. They don't give away Nobel Prizes. And as they did to Sir Hodgkin here, Dr. Hodgkin, who won the Nobel Prize. Whoops, I don't think I wanted to do that. Whatever it was I just did, I don't want to do it. What year was that? Oh, geez, let's see, he died in 1998. I want to say it was in the early 60s, late 50s, somewhere in there. Anyway, that's the Hodgkin cycle. Not even sure it's in your textbook anymore. But, and so pictorially, that's what it looks like. You have electrically gated sodium channels at threshold being opened, more sodium comes in, and it just propagates along the long axis of the axon. Of course, if this were to occur, if the trigger zone were to occur somehow magically in the middle of an axon, it would propagate in both directions, right? But this is, we've stipulated that we're starting at the graded potential dendritic end, and we've reached a trigger zone, and now there's only one way to go, and it's down. That's not the same Hodgkin, by the way, as Hodgkin lymphoma. Somebody chatted. That was the Hodgkin of lymphoma. Was another famous Quaker, actually. I don't know if I suspect they were related, but he was a century earlier. Yeah, almost a century earlier. He described this disease of the lymphatic system, Hodgkin's disease. Anyway, and you have this self-propagating wave going down the axon. And we've got refractory periods and potassium channels opening, and then the sodium-potassium pumps doing their thing. And there it is, all of that in one tidy diagram. It's too much, but there it is. So that's all review. And so if you were looking along any point in the axon, you would see this action potential propagate. So this is distance now on here, not time, but distance along the axon. And right here is where the sodium channels are opening, and boom, you have this change in membrane potential. Over here, things are closing, and the sodium-potassium pumps are reestablishing membrane potential. Down here, it hasn't happened yet. And so because there's this refractory period, that is, they opened and then closed immediately, once it's occurred here, it can't go back that way. That's kind of an important feature. That is the discrete nature of these sodium channels that open for a brief instant and then for another brief instant can't open again, means that once they've opened, it can only go one direction. It's not going to just sit there and cycle back and forth, which was, you kind of defeat the purpose, I suppose, of an action potential. We probably wouldn't call it an action potential anymore. We would call it a inaction potential. Anyway, this is moving along that way, big arrow. And you can imagine this in a perfect world, that if you were to then to measure, put electrodes anywhere along this axon, you would see the exact same voltage change at each of these points. And that is, this could go on indefinitely. However long the axon was, it would continue. That's in a perfect world. Of course, the world isn't perfect. And I don't know if this is a picture from your textbook. I think it isn't. It's a picture from somewhere. And the picture itself wasn't even perfect. It was trying to represent the imperfect nature of axon transmission. In the process, blew it. But what it really means to say is this. And remember our degrading current that we saw so nicely inside of the dendritic end? Well, that can occur anywhere. Plasma membranes can leak wherever they are, even on the axon that is supposed to be transmitting this action potential. And there are various phenomena that can get in the way of transmitting this action potential longitudinally. We want this voltage change to proceed from one end to the other. And the faster we can do that, the better. Yet, all along, there are going to be roadblocks. There are going to be problems. There's going to be entropy. We're creating voltage changes that require establishing concentration gradients of ions and packing energy into one place or another. And entropy is always going to be combating that. And this idea of the imperfect nature of the plasma membrane gives rise to something called the length constant, which is how far away, if you were to stimulate this axon right here to some maximum voltage, how far away from that point would you get before you had a 63% drop in the maximum voltage? That is the definition of lambda, the length constant. And so what that's telling you is that how far away along this axon are you going to lose that voltage that you need to propagate, to self-propagate? Obviously, if you had that big of a drop and it dropped that far and it dropped below some voltage that was necessary to open the next voltage-gated sodium channel, then the action potential would stop and you have a problem. So in principle, we want a really, really, really long length constant, which means that the membrane or the axon is really holding the charge well, and that means the charge will propagate well. So bigger length constant, better. Shorter length constant means that things are dropping off too fast. You might, some of the phenomena that to which I was alluding, one of them is capacitance. So imagine you have here, so this is a plasma membrane drawn as a circuit diagram. So here's the channel, the ion channel, which would have some resistance. In the case of sodium channels, not much resistance at all. In other words, once those babies are open, the sodium is going to flow through there like crazy. So that's a good thing in this case, because that's going to create ultimately our flow along the long axis. But another part of this circuit diagram here is a capacitor. And you'll remember from physics, capacitors are these plates that hold charge. Can hold very, very, very large charges. That is, one side will be positive, one side is negative. And you can get these things really close together and they can sit there and hold charge. Very limited. I mean, you can only release it in a burst. And they're very useful that way, a capacitor. You can start big electrical devices with a capacitor by simply releasing the potential in that capacitor. And how they work, of course, is that you have, in the case of electrical capacitors in machinery, you have electrons built up on one side and not the other. And the positivity on one side holds the negativity in the other. And they're just attracted to each other, but they're just so, because they're really close together, but they're far enough apart that no flow happens and you hold a charge that way. Well, the same thing happens on a plasma membrane. Remember, we have sodium is built up out here. And when at any point, if you were to look at the sodium positivity here and the relative negativity here, and then we're to try to move sodium ions, because this plasma membrane is pretty small, that is, the distance here is very small, 5 nanometers. That's plenty close to allow charged molecules to attract each other. And so because of that attraction across the plasma membrane, they don't want to move, which is a problem. You're not going to get flow of any kind if there's a lot of membrane capacitance. So ideally, if you want, I don't want to look at that one. Let's go look at this. That's too many. There you go. Here's the complete circuit diagram here. So all along a plasma membrane. Here is, where's my cursor? There it is. The outside, no, jeez, I'm sorry. Yeah, inside, cell interior over here, cell exterior. These symbols represent capacitance. These symbols represent the membrane resistance. The red arrows here, let's say we're starting an action potential down here. We need to move ions this direction. Hindering that movement will be, what if ions wanted to go through the plasma membrane? You would lose then the flow this direction if they went this direction. If they go this direction, they might go this direction because they're locked up in capacitance right here. Or they might go in this direction if they can actually flow through a leaky plasma membrane. That would be bad too. Anything that detracts from the ions moving this way is going to lose our action potential, decrease our length constant, and decrease the efficiency and speed of this action potential. So that length constant mathematically just looks like this. It's the decay of voltage, and it's just a simple exponential function, natural logarithm type exponential function. And x is the distance from current injection, lambda is the length constant, and so e to the negative x, because we're going away, over lambda is the exponent. All of that ends up at this. When x equals lambda, that is the distance equals the length constant, then that is a 37% of the maximum voltage that you saw somewhere else. That is, it's decayed by 63%. So what determines this lambda? What makes the lambda large or small? And you don't need to know this equation or anything. I'm not going to ask you to calculate anything. It's just if you're wondering where of what significance the length constant is. Well, this equation, on the other hand, has actual variables from the membrane potential that matter. Rm is the membrane resistance. Ri is the internal resistance. Ro is the external resistance. So going back to our picture, here you have a certain amount of resistance going this way. That's with these little resistors here. So there's a resistance going this way. We want the flow to go this way. We also have resistance here, right there. That's the resistance to losing ion flow out of the leaky plasma membrane. And then for this to be a complete circuit, we have to have a corresponding backward movement of ions this way. And so there has to be resistance out here as well as flow out here. Now, outside the cell, there isn't much. Inside the cell, we've got we've got the simple space restrictions of the size of the axon. We've got the microtubule network, the cytoskeleton. We got all those motor proteins moving stuff. There's all kinds of crap on the inside. And so the bigger that axon is, the less resistance there's going to be longitudinally on the inside. The outside is pretty uniform. pretty easy, not a lot of resistance out there. But go back to that equation. If the membrane resistance is big, lambda will be big. If the longitudinal resistance, either inside or outside, is big, length constant goes down. You want these to be very, very small. You want that to be very, very large. That will result in a large length constant. That is, you'll be able to go a long ways down the axon before you get a 63% drop in voltage. There is that again. Where was my capacitance picture? I don't know. I thought I had another capacitance picture. Anyway. So what can we do to make sure the length constant is as large as possible? Axons that are any significant length, that have to go more than, say, about this far. I mean, we're talking centimeters here. Small, 2 or 3 centimeters. Probably even far less than that. Most of them would be what we call myelinated. Is that word? Yeah. Here's the myelin sheath. Myelin. That is these axons, to increase their length constant. There is a cell called a Schwann cell, it's on the next slide, that has a particular protein in its plasma membrane called myelin. It wraps around these axons and thus insulates them and vastly, vastly changes their length constants. They change their capacitance, they change their membrane resistance, and in so doing, greatly facilitate the efficiency and efficacy, that is the speed of action potentials down long axons. It works like this. So this myelin sheath is going to have open places as you proceed down this axon. These open spots are called nodes of Ranvier. And the nodes are simply open spots. I'm surprised that that guy's name is still there. It's the person that discovered them. Those nodes happen to be open spots where there's no myelin sheath of Schwann cells. It also happens to be where the preponderance of sodium, voltage-gated sodium channels reside, as well as the sodium-potassium pumps. So what happens here is that the sodium comes in. So let's just use these two. Sodium's coming in here. These are voltage-gated sodium channels. They've got tripped by a voltage change coming in down the long axis of this axon, and they got opened. Sodium comes in. If you imagine this spot right here, beneath this myelin sheath, imagine there aren't any sodium-potassium pumps here. So in other words, this part of the cell is essentially polarized in the opposite. There's a lot of sodium in here. So when you add sodium here and add positive charge, what you tend to do is, well, imagine this is the analogy a friend of mine used, and I kind of liked it. Imagine you have a piece of pipe, and it's got ping pong balls in it, about smaller than the pipe, and so it's full of ping pong balls. And this is the second time I've used ping pong balls as an analogy. That's deeply suspicious. Anyway, the ping pong balls enter the pipe here, and as they do, there's already ping pong balls here, so they displace this direction, and that charge that the ping pong balls, sodium ions, carry moves to here and then opens these. So in other words, these sodium ions that enter here don't actually have to move all the way to here. They actually just are able to transfer their electrical charge, their electrochemical energy, through the lots of sodium ions that are here, all the way to this next node. That is, the current actually jumps like a little teleporter from one spot to the next, which is a major time saver, as teleportation tends to be. That would be bad. I've decided that teleportation would be bad for us. I don't want anybody to invent it. And it goes beyond just, say, the problems that Jeff Goldblum had in The Fly. If you haven't seen that movie, that's your homework for the weekend. Anyway, this teleportation speeds the conduction velocity of the action potential many, many times. Look what would happen without the myelin sheath. Sodium enters here, except without a myelin sheath, the sodium ions would simply leak out here, as it turns out. The myelin sheath acts as an insulator. It prevents leaking sodium channels. That is, it increases R sub m, membrane resistance, manyfold. So as you make membrane resistance bigger, your length constant gets bigger. Let's see. So we've increased membrane resistance with those things. And here's the myelin sheath. There are two types of cells, as it turns out, that form the myelin sheath. It depends where you are in a vertebrate body. In the central nervous system, they're called oligodendrocytes. In the peripheral nervous system, they're called Schwann cells. Whatever. They're very similar. They do exactly the same thing. They have a protein, myelin, sphygmomyelin, in their plasma membranes. And I think I have a better picture of that. Maybe not. Imagine a balloon. I mean, really, a rubber balloon. And you're going to take a rubber balloon and you're going to wrap it around a bare wire. And you take the rubber balloon, and of course it's closed, it's a bag, basically. And the rubber is the insulating portion of it. And then you got all the cellular stuff going on the inside of the balloon. But then you wrap that thing around, and it wraps itself around and around and around and around and around. And in so doing, you get many, many layers of that rubbery plasma membrane, which is the myelinated plasma membrane, around the axon, and thereby insulates it. It increases membrane resistance manyfold. If you want to understand just what this does for the velocity of propagation, what we call this jumping from one node to another is called saltatory conduction. Saltatory is jumping locomotion. Saltatory conduction, in this case, increases the speed of conductance massively. Just as an example, what happens if you were non-myelinated? And as it turns out, all of the early work, and a lot of work still, is done on squid neurons. Hodgkin's work was on squid neurons. And the reason squid neurons are useful is that they're enormous. And so you're trying to put probes in axons, and these are really small things, except squids are enormous, a couple orders of magnitude bigger, which, with the primitive instruments, especially, back in the 30s and 40s, this was a big deal. And so if you were to look at conduction velocity on the y-axis here, and this is the actual, so the diameter of the squid axons goes from 0 to 800 micrometers versus A myelinated cat axon is but 3 micrometers or 6 micrometers, 2 orders of magnitude smaller. But the difference is, and the squid conduction velocities are pretty fast. When you get to the big ones, they're up here close to what they are in a cat, except a cat achieves those conduction velocities with an axon that is 2 orders of magnitude smaller than a giant squid's. So think about that. Now, why is it that a squid has why hasn't myelin occurred in the squid? Some of these squids are really big, really big. And so why wouldn't you want myelinated axons in a squid? Or the other way of thinking, why do you need them in a cat? Does velocity have anything to do with reaction time? Yes, it does. Obviously, the action potential conduction is going to be a rate limiter when it comes to how quickly you can, say, have a reflex arc occur. I'll talk to you about the recording. It'll get ended up posted on Canvas. So short version, non-myelinated squid axons, squids can get away, squidly diddlies can get away with large axons because, well, let's think about your spinal cord and all the myelinated axons there are going down your spinal cord. The whole thing is as big as my thumb. Hundreds and thousands of neurons. Size of my thumb, with all those neurons. If I were a giant squid, that thumb would have to be 2 orders of magnitude larger, 100 times bigger. That's hard to imagine. So in other words, my spinal cord is the size of a trash can lid diameter. Now, that's not to say that you couldn't make an organism that had a spinal cord that large, but imagine such a big mushy structure as a neuron, which they are, subjected to 1 g of gravity all the time. And what that would do to just the mechanics of the animal. That's a lot to carry around. For a squid, it's nothing to carry around. It's in water. Gravity is essentially irrelevant. So they can get away with giant axons because they don't have to carry them around. For terrestrial animals, though, having myelin and making as a result, your neurons, your axons can be far, far smaller, is a huge space saver, which means it's a huge mass saver, which means it's an enormous work saver because you're not carrying it around against gravity. So we're all happy that squids don't have myelin and very large axons, because we wouldn't know nearly as much about neuron physiology as we do were they not big and accessible to experimentation. Go on. Oh, here's some pictures of, aren't they cute? They look like little marshmallows wrapped around in these pictures. Very nice. I'll come back to that picture in a little bit. I think I'd use it again. There they are again, the Schwann cells. They decrease membrane capacitance and increase ion flow 5,000 times. The capacitance thing, which the slide has just disappeared, I don't know where it is. But again, think about, so here's your plasma membrane, and you've got a bunch of sodium ions now on the inside, because that's the inside, and they've flowed in. and now it's negative out here. Well, if this distance here is just too small, then these sodium ions are going to be locked up with the negativity out here. And so they're not going to want to move this way. They're going to be latched in place. So if instead they're all drawings, if instead you have a plasma membrane that has, here's our cell, if you have a plasma membrane that is insulated with a Schwann cell, and now when sodium comes rushing in, any negativity that's out there is this far away. We've increased the distance here. And as you increase the distance, you exponentially decrease the attraction of these ions, which allows longitudinal flow of the sodium ions. Without that longitudinal flow, we don't get a propagation of action potential. So the combination of decreasing capacitance and increasing membrane resistance to flow back out, eliminated by, again, the Schwann cells, means that increase longitudinal ion flow 5,000 times. In other words, the action potential is 5,000 times faster than it would otherwise be. Pressure does not have the same effect, or is it because it switches up all sides? I'm not sure what that's referring to. Not sure what that
Are you talking about the water pressure question?
What water pressure question? Yeah, yeah.
She was talking about
in the squid. Yeah. Well, water pressure is essentially what's holding the thing up, right? It has its buoyancy. So a squid, there's no effect on ions so far as anyone knows on say a giant squid that's down at 1,000 meters. Increase in water pressure, there's no effect at the ionic level.
I have a quick question. Is the myelin sheath, is that just a cell membrane with a lot of sphingomyelin in it?
Yeah, exactly. It's just, yeah, that's what you're looking at is this. You can kind of see it here. So here's the nucleus of the actual Schwann cell. And so that would be crammed off on one side of the cell. And then imagine the rest of the cell just wrapped around. So you've got multiple layers of the plasma membrane, which is studded with the sphingomyelin all around this axon, which just insulates the crap out of it. I mean, it's utterly impermeable. And the capacitance goes, essentially, it's almost zero. And then every 1 to 3 millimeters, you have a node of Ranvier where the ions can actually flow.
Did you say that it was one Schwann cell per in between the nodes of Ranvier?
Yeah, well, each myelin it's kind of like it's like a maple bark. Nah, it's not a maple bark because that has something going through it. It's through its middle. Each one of those Schwann cells is one cell that creates that myelin sheath in one spot, and then there'll be a gap, and then there'll be the next Schwann cell.
Just another quick question. So
yeah.
The point of decreasing the membrane capacitance is so that the charge from the action potential doesn't get absorbed by the membrane and lose its power. So decreasing capacitance just it essentially keeps the action potential from pittering out.
Yes, well, so you have to think about the longitudinal resistance, right? So we want once the sodium comes in here, I'll go to this picture. Once the sodium comes in here, I want a different color. It has to go this way, right? And again, it'll jump then. But any kind of it requires some longitudinal movement. And if instead it just entered and got locked up by the negativity, if these Schwann cells weren't there, you would get no longitudinal movement of ions. That little saltatory jump would never happen, because the ions would all be, the positive ions in here would all be locked up with the relative negativity on the outside of the cell. And so nothing would move. The cell membrane would just be a giant capacitor, and everything would just go, and that's it. So the Schwann cells are increasing membrane resistance. That is, they don't allow ions to leak back. So increasing R sub m, and at the same time, they're decreasing the longitudinal resistance inside, because by the loss of capacitance on the cell membrane. So when you increase longitudinal resistance, decrease longitudinal resistance and increase membrane resistance, you get an increase in length constant. Okay, so you don't lose any charge from here to here. That length constant is so big because of the myelin sheath. If you were to measure length constant, it would be, you wouldn't see virtually any decrease in voltage from here to here. If those weren't there, you would see that decrease in voltage from one point to the next, and it might not even be enough to propagate the action potential.
Okay, that makes sense.
Okay. Here are all drawings.
Professor?
Yeah.
When a sodium ion passes its energy to the next ion, when does that, or where does that sodium ion then leave?
And so, well, you can imagine, if you want to think of it this way, they're coming this way, going this way, and then there's sodium potassium pumps here. So they'll get pumped out. These would get pumped out. You also have flow on the outside here going this way, because as this became negative here, because sodium ions came here, there's positivity out here. So you have to think of this as a conga line. Conga, do they still do that, things like that? Conga lines where everybody's in a line and you're dun, 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 dun. And if you move, the person in front of you has to move, and the person behind you has to move. So anytime you're thinking about ions or electrons, there can be no just gap of ions or electrons. If these move this direction, then some out here are going to have to move this direction, because it's all on a chain. It's a circuit, literally a circuit. So does that
can reach his pocket?
Does that answer the question?
He can reach his pocket.
Stand by. Yeah, that was not a question. Did Riley? Did that make sense?
Yeah, that makes sense. So just to reiterate, the voltage-gated sodium ions pump them in, and the sodium potassium pumps get them out.
Yeah.
Awesome. Thank you.
Yep. Oh God, I'd forgotten I had this, and this is creepy. Not that, you're probably fairly callous to creepy right now in terms of microbial disasters, but this is a scary one. When I was an undergraduate, this is 1979 or something, I had a professor who alluded to this, and I have never been sure that it wasn't just an urban legend, or more properly a Cold War legend, because the Cold War was way on in those days. And I grew up with bomb drills. You would duck and cover and stuff like that. I'm sitting in the middle of Nebraska, where we had, Minuteman missile silos out in the countryside. And when I heard this story, it was even scarier than the nuclear holocaust. The Soviets, back in the 50s and 60s, came up with what they thought was a very clever, and I suppose it is, from a purely aesthetics of function point of view, a quite beautiful biological weapon. That is, they engineered, I think it was a bacterium that's in here somewhere, yada, yada, yada. Yeah, it was some kind of bacterium. Oh, there it is. That genetic L. pneumophilia. I'm not sure what that bacterium is. They engineered it to, that is, they gene spliced that bacterium with myelin. And so, and then they would inject it. This is the testing phase of the story. They would inject that into a bunny. It's always bunnies, isn't it? And the bunny's immune system, then in responding to this bacterium, said, oh, invader, your immune system works on the principle of recognizing foreign proteins and says, must kill the bacterium with this protein. And so it does. The problem is that your immune system then is unable to distinguish between the bacterium carrying that myelin protein and your body's own myelin surrounding your axons. And so then the immune system of these bunnies would attack their own myelin sheaths and would, well, that essentially you have a bacterium that is creating multiple sclerosis. That's what multiple sclerosis is your immune system, for reasons that are still unclear, sees your Schwann cells as the enemy, and your immune system starts killing them. And in the end, you have no myelin sheath to your axons, which means, in particular, your long motor neurons that really require myelination to function properly don't function properly. And they stop being able to transmit action potentials effectively or fast. And the lack of muscular coordination, muscular activity, well, you've seen it. One of my very best friends from high school, he's a physician, but for all of his adult life, he's been battling MS. Went from one of the most athletic people I've ever known personally to, well, he gets around the hospital on a Segway now, which is kind of cool, but he can barely walk. Devastating thing. And the Soviets were actually thinking about creating a bioweapon that would do that job for them. So it wasn't made up by some professors, as they tend to do. So anyway, what happens in a non-myelinated neuron, you could still get an action potential to propagate, but think about the number of spikes, that is, electrical depolarizations that would need to occur at each stage down an axon. And compare that to the number of such depolarization events that would occur down a myelinated axon. This thing is going to go, there's an animation associated with this, but it's silly. Imagine the cycle of sodium entering and getting pumped out. So essentially what you have is, the sodium movement is going, and you get action potential and it's propagating. Yeah, we're getting there. Yeah, we're getting it's the Pony Express. That was back when they delivered mails on horseback. The Pony Express. Without myelin, this is the Pony Express. You'll get the letter a year later. That's assuming the writer doesn't get killed on the way. That is leakage or some other bad thing that could happen. Versus this is just boom, 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 done. It's many, 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 many times faster. That allows the coordination of neurons. I have neurons that are sending action potentials from my motor cortex up here all the way down to the base of my spine. They're this long, and they have to coordinate with other neurons that might have much shorter distances to go. But because everything's happening so fast, it's still possible to coordinate them. This is extremely sophisticated stuff, and speed is everything. If you want to be, there's no point in reacting if you react slowly. I'm not sure what that was referring to. Okay. So, what happens when we get down? Everybody, any questions on action potential propagation?
Yeah, just a quick question on saltatory conduction.
Uh-huh.
So it's not literal teleportation like you were saying earlier. It's more just
no, it's not. No,
it's more just depolarization occurs at the nodes, and that's enough of a voltage change to trigger the next node down the line to open.
Yeah.
Okay.
If you want to think about it as energy teleportation, have you ever seen a Newton's cradle?
Oh, okay. So yeah. Energy teleportation. That makes more sense.
And the other one at the other end goes out. Yeah.
Yeah.
That.
Yeah. It's just energy
Maybe that's
teleportation,
a better
not literal teleport. Okay. Yeah, that makes more sense.
Okay. So what happens when we get down to the end? We got the action potential going down. It gets down to the terminal end. Let's say a motor neuron going to a muscle cell, or to another neuron. You could have a synapse where you're talking to another neuron. That happens a lot too. In that case, you would have a presynaptic neuron and a postsynaptic neuron. So we'll be referring to these in terms of where they are in the synapse in the chain. In between neurons, you could have a simple electrical synapse. Electrical synapse. Somebody's talking about the thymus cells and T cells. Jeez, little subculture of chat over here. In the electrical synapse case, here you have two neurons, and that action potential is going to come down. And the simplest way to transmit that action potential to another neuron, I suppose the simplest way, would simply be have a physical connection between the neurons and just let the sodium charge continue on through. That happens in electrical synapses. And there are a lot of electrical synapses of neuron to neuron. You just have gap junctions between the plasma membranes. You get sodium ion flow, boom, and you have a transmission. Of course, you would be going from one neuron to the next, and that next neuron, the postsynaptic neuron, the sodium charge would be entering the dendritic end, so it would be a graded potential at that point, and so it would have to be big enough to reach the trigger zone of that postsynaptic neuron. This happens frequently in neurons that are in the central nervous system of the brain that are very closely packed, and essentially it causes neurons to function as one. If one does this, the next one will always do this as well. And when you have circuitry that requires the integration always of several neurons, this is a very efficient way to go. So gap junctions, you've seen those before, and you have a simple electrical synapse. One to the next. Okay, which way is this going? I think it's going this way and this one. Okay, that's pretty easy to imagine. Nothing terribly inspired there. The question of whether all synapses behaved in that way was a question that went on for a very, very long time, hotly debated for a very long time. And here's another person, Otto Lowy, and his contemporary, Henry Hallett Dale, a couple of eminent physiologists in the late 19th, early 20th century. I guess the story goes that Lowy dreamed the experiment that would prove that not all synapses were electrical. He was struggling with this problem of trying to show that there was some other kind of synapse that is a chemical synapse, which had been hinted at, but nobody could show it. And he went to bed all pissed off that he couldn't come up with an experiment, and he dreamed the experiment. He said, "Oh, wait, what if I put a neuron with a muscle in just a neuron, sorry, not with the muscle, in a petri dish and stimulated the neuron and then took the neuron out. And so if it was chemicals released by the neuron, if I then put the muscle in that petri dish that had now the chemical of that neuron released, the muscle would contract and it would show that there was chemistry, not charge involved." And he dreamed that. That's what he says. Yeah, I came up with my the answer in my sleep. Has anybody ever done that? You've been struggling. You got too tired, overtired. You were thinking about something, and you finally said, "The hell with it, I'm going to bed." And as you were sleeping, you dreamed the answer to the question. I've done that too. I thought it was made up, but it's actually happened to me. That's kind of scary, just how rejuvenating and physiologically important the dream state must be. It's your brain gets stuck. 2 + 2 = drool, you go to sleep. 4, it's 4. God, what the hell is wrong with me? Yeah, well, that's what happened to Otto. He proved the chemical synapse. On a chemical synapse, it goes like this. As the action potential gets down to the terminal end, what it does is cause voltage-regulated calcium channels to open. Here's calcium again. Calcium is the ultimate signaling ion. It's always involved. Calcium channels will open. The calcium enters the cell and causes conformational changes in a bunch of proteins in there, we'll see them here in a second, that ultimately causes synaptic vessels full of neurotransmitter to fuse with the plasma membrane and release neurotransmitter. Here it is represented as a diagram. There's a lot of moving parts here. So here's the presynaptic neuron here, and say here's a muscle cell. Since we've already talked about muscles, easy one to remember. So here we get down to the terminal end, and you don't see the calcium coming in, but I'll just stipulate that there are voltage-regulated calcium channels open, and the calcium comes flooding in. And what the calcium does is binds to, in particular, a protein called synaptotagmin. And synaptotagmin is inside the plasma membranes of these vesicles, which then causes synaptotagmin to bind with this collection of proteins called snare proteins. There's synaptobrevin and SNAP25 and syntaxin. And those all bind together with ultimately causing the synaptobrevin of the synaptic vesicle and the synaptotagmin associated with it to bind to these guys, which pulls the synaptic fusion complex down to the plasma membrane, and it merges with the plasma membrane, and the neurotransmitter is released. Gee, that seems, that's quite the Rube Goldberg machine. That is, seems really complicated, and obviously it has to happen a lot faster than the period of time it took me to describe it, but it happens, and every time my mouth is moving, that is going on because we're releasing acetylcholine onto muscles that are moving my jaw and tongue and controlling my vocal cords and my gestures. And our temporal reality is just not properly equipped to understand how quickly these things happen. The acetylcholine goes out, and ta-da, on the muscle cell in this case, we have acetylcholine binding to chemically gated sodium channels, and they open. And you get an action potential across the plasma membrane of the muscle cell. By the way, these proteins right here, these snare proteins, are unusually susceptible to being chewed up by toxins released by microbes called Clostridium botulinum, the gram-positive bacteria that are responsible for botulism. Botulism, these are facultative anaerobe bacteria that work well, grow well inside, say, a can of improperly stored tuna or something, or beans. And before we had proper canning techniques and people were creating anaerobic environments, well, they did it at home. You know, home canning was a big deal, and still is. And done properly, it's entirely safe. But the reason that you boil stuff before you can it is that if you leave any Clostridium inside this can, which will now have this perfect anaerobic environment, they're just going to go crazy. They're going to, oh, hey, somebody nicely got rid of all the oxygen for us. Let's make babies. And when they make babies, they make toxin. And this toxin is extremely powerful. It gets into your neurons. It degrades your snare proteins, which means when it comes time to, say, do something important like breathe, your motor neurons to your diaphragm that are going to allow you to ventilate say, go, go, gadget, diaphragm, and the action potentials will be sent down, and they'll get down to the end, and the calcium will come flooding in, and the snare proteins are all just busted up, and nothing happens. The muscles don't contract, and you suffocate. Bad way to go. It used to be that botulism was was a fairly significant public health problem in the early 20th century, late 19th century. And of course, leave it to the enterprising humans out there. They said, hey, you know, that's a really powerful toxin. I wonder if there's anything cool we could do with it. I mean, aren't there occasions when we really would like to prevent muscles from contracting? Oh, by the way, here's synaptotagmin here somewhere. There it is right there. There you see the calcium ions binding to it. There you go. Oh, more pictures of it. Yeah, they're finding out more and more about synaptotagmin and how it binds calcium. You don't need to know any of that, but it's just for fun. Let's say you wanted to, for some reason, say you were a person who is unhappy with the lines that your muscles in your face were creating. That is, those muscles that create various facial expressions. Expressions that were the bread and butter for, say, actors. So I'm not exactly sure how they became preoccupied with it. But if those muscles that create facial expressions are also those facial expressions that you make, then you laugh a lot and you squint, and when you're laughing a lot, you're squinting because you're laughing, and those muscles that are squeezing your eyes are creating little crow's feet, which apparently our youth-obsessed society has decided are ugly. Well, if you simply, if you paralyze those muscles by injecting small amounts of Clostridium botulinum toxin in there and destroy the neuron's ability to release acetylcholine, then you'll paralyze the muscles. You won't be able to squint and create crow's feet. And so the lines that indicate age and experience, and in some cases wisdom, but not all, will not develop. So here you see the results after Botox. That is Botox. Here's the person gets injected here and up here, and then after a certain length of time that those muscles have been unable to contract, that is, after a certain length of time, this woman has been unable to express surprise or anger, then then the lines, yeah, sure as hell, they go away after a while. Okay. If anybody understands this, email me. I've been wanting to understand it for years. I've tried and tried and tried. It's just another one of this huge file of things about society that I don't understand. But if anybody has any revelations, please share. Meanwhile, the ligand, yeah, we've got that, and the channels of greater potential. And the neurotransmitter, as we saw before, acetylcholinesterase degrades the neurotransmitter in this case, and the interaction stops. And we've seen all of that. That's with our simple muscle model. That one's we've been over. That is what we would call a direct effect. That is, we have a neurotransmitter comes down, it binds to a sodium channel, and opens that sodium channel. Ions flow. Direct, nice and simple. It's discrete. It happens and then it stops happening. And the effect is extremely short term. Direct. There is also, and that's an important thing, that's happening in all kinds of neurons, especially muscles, interaction between motor neurons and muscles. There's another category, if you will, of postsynaptic effect called indirect. And in those cases, indirect effects, the ligand hits a channel here. So here's the channel. Here's the ligand coming down. It binds to a plasma membrane protein and then sets in motion usually a chain, a sequence of membrane events that ultimately produce large changes in the cell, in the postsynaptic neuron, in this case. You can turn on genes, you can change the behavior of the postsynaptic neuron. This is a really important effect. Obviously, when you can have an action potential that reaches, and this is always in a neuron, a postsynaptic receiving neuron. If you can actually change the behavior of that recipient by simply talking to it, then this is a very powerful mechanism for things like memory, for example, where you create neuronal pathways that you constantly travel down. And I showed this once before, where we're taking GTP, or before, this is where we're interacting with GTP and adenylate cyclase, for example. And we'll talk about specific examples of this pathway occurring. You're going to see it happen inside the neurons, the receptors of the eye, for example. And in the end, you can create what they call a second messenger response, cyclic AMP. And remember, these are things that can tend to amplify. So for the price of one ligand, we can turn on enzymes in here that can produce profound changes in the physiology of the receiving neuron. So this is a way of altering pathways and that is potentiating neurons for a very long time. That's now those behaviors that occur in the postsynaptic neurons can basically be one of two types. You can either be excitatory or you can be inhibitory. So we'll talk about excitatory postsynaptic potentials, where you have, as we've described before, opening sodium channels and actually causing further action potentials in the postsynaptic neuron. Or you could also have inhibitory effects, where the neurotransmitter gets to the postsynaptic neuron and says, no, I don't want you to behave. In fact, I want you to decrease your activity. In those cases, that neurotransmitter might go out and cause an increase in potassium or chloride channels permeability, and thus hyperpolarizing rather than depolarizing the dendritic end of the postsynaptic neuron. They'll actually shut it down and say, no, we don't want you to respond. We're going to inhibit you. We've been talking, and naturally it's natural to speak of exciting one neuron to the next, but equally important, absolutely equally important, is the ability of neurons to inhibit other neurons. Because otherwise, everything's connected to everything in there. You can always get somewhere from over here to over here. If you're thinking about the billions of neurons in a human brain, there's always a pathway you could get there. You don't want every pathway to interact like that. You want to be a little more discreet in the way you turn things on. And so that requires that you eliminate in a transient way, eliminate some of the pathways so you get the discrete effect. Otherwise, your head explodes, basically. Everything fires, and you have things like epileptic seizures and things like that, where you have chaotic firing of neurons. So we'll talk more about inhibition in a little bit. Neurons can, in their effect on other neurons, produce postsynaptic effects that are either a result of temporal or spatial summation. And by that, I mean this. Here is a postsynaptic neuron right here, this typical alien one. And here's the presynaptic neuron that's coming down, the green one. And in this case, it's going to stimulate it. And if it stimulates it and its stimulation is inadequate to be threshold, to meet threshold on this dendritic end, then you get nothing. And then it does it again a little while later, and still nothing happens. But if it were to stimulate it twice at a higher frequency, that is, it hits it here and then tries to stimulate it again before it gets all the way back down to resting potential, then those will summate. And between the two of them, it'll rise above threshold and you get an action potential. That is temporal summation. It makes sense. If you go bang and then wait, bang again, nothing's going to happen. But if you go whack, whack, in quick succession, those stimuli will summate and get you over threshold on the dendritic end, and you get an action potential. So that's temporal summation. Temporal summation can happen from one neuron just repeatedly bugging the postsynaptic neuron. Spatial summation is when you have two neurons ganging up on a postsynaptic neuron. And in the same way, while neither of them alone might be enough to stimulate the postsynaptic neuron, if they both work in concert, they can summate to threshold and get an action potential in the postsynaptic neuron. So spatial summation there. Here you could have spatial summation that cancels each other out. This one's trying to stimulate it, this one's trying to inhibit, and it's a wash. Nothing happens. That's also a kind of summation. It just sums to zero. Further, you can have, as I mentioned before, with these indirect mechanisms, you can potentiate neurons. And this is a really important effect for creating things like memory engrams, that is, these neural pathways that represent concepts, words, memories of any kind. This potentiation is strict definition is increased activity at a presynaptic neuron results in increased postsynaptic graded potentials. In other words, with every presynaptic neuron attempt at stimulating a postsynaptic neuron, with each attempt, the postsynaptic neuron starts to increase in its response. In this case, it's just the opposite of what we thought about before, where we were thinking about a negative potentiation, where a neuron's threshold starts to increase as you slowly are stimulating it. In this case, this kind of potentiation is exactly the opposite. The postsynaptic neurons start to really sit up and start paying attention and actually increase their responsiveness with an increase in stimulation from the presynaptic neuron. Here's an example of it. This is a kind of a cool experimental setup where you have two neurons here, both talking to this one. And these are identical neurons. They call them collaterals. This happens all the time in the brain, where there's essential redundancy in neurons talking to other neurons. And so you have identical neurons here. And this one over here, stimulus 2, we only stimulate it once, boing, and the response of the postsynaptic neuron is this. Versus this one, we hit it with a tetanic contraction. That is, we stimulate it really hard and then pause for a second and then hit it again. And when you hit it again, its response is far larger. It went from 100 to 300. It trebled its responsiveness because it got stimulated, it got kind of spooled up beforehand. So that's a kind of potentiation. You can also have the exact opposite, of course. As I said, it's just as important to be able to inhibit neurons as it is to stimulate them. Otherwise, it's just a complete catastrophic, disorganized mess of neural activity, which I find myself, a point at which I find myself every day at about 4:30. It's right during recitation, so that explains everything, doesn't it? You can have synaptic pruning, and you can have what's called neuromodulation. Synaptic pruning, you've seen this slide before. And synaptic pruning is just saying, well, we can attach these neurons in a variety of different ways. You could have a synapse coming in, talking to a dendrite here. You could actually have a synapse right on the cell body itself. You can skip the dendrite end. You could have a neuron that's attached to the trigger zone of a postsynaptic neuron. Obviously, this one, in connecting it in a more traditional way, is going to have to work through a graded potential through the dendrite. But this one, on the other hand, can skip all the graded potential of the dendrite, would potentially be far more effective at doing either stimulating or inhibiting. Or you could have right here, let's say this one right here. Let's look at this axonic, as they call it. That's just one axon talking to another axon. In this case, right at the trigger zone. Imagine this one saying, no, I don't want your trigger zone to respond. So this one might be trying to stimulate it, but this one right here is causing potassium channels and chloride channels to open, and it's getting way negative on the inside of this cell. So negative that a graded potential that would otherwise cause the trigger zone voltage to go up past threshold instead doesn't make it. So here's our graph of voltage. And so it's going along and you're hyperpolarizing it. So it's way down here. Instead of -70, it's down at -90. And threshold is here, which means a graded potential is going to have to not just go this far. A graded potential is going to have to be big enough to actually increase the voltage that much. That is, this axon, this trigger zone has been inhibited and is less likely to fire as a result of this synapse right there. Really important, happening right now all over the place in your peripheral and central nervous system. Yeah, I need to talk about that. And finally, last thing, as it turns out, we'll talk about hormones later in the term, that neurotransmitters are not the only ligands out there. Turns out that there are a ton of hormones, say this one right here, that's GnRH, is also known as gonadotropin releasing hormone. We'll talk about it later. But that is a hormone that stimulates the gonads. And as it turns out, it also has powerful effects, neuromodulation effects, and can very much change the behavior of ion channels. And so hormonal effects, they're real. Is PMS a real thing? Yes, it is. Neurophysiologically, absolutely, changes in hormone levels will change the behavior of neurons. So we'll see a lot more of that later. Any questions? Happy to hang for a minute if you have any questions. For the rest of you, I'll see you Thursday.
I think I missed when you discussed the difference between pre and postsynaptic. Is the presynaptic just doing the stimulating and inhibiting while the postsynaptic is being stimulated or inhibited?
I'm sorry, run that one back again.
I'm just wondering about what the difference is between pre and postsynaptic. Is the presynaptic neuron the one doing the stimulating or inhibiting, and the post is?
Yeah.
So
the synapse is the connection, the space between them, right?
Gotcha.
And so whatever is before. Yeah.
So does that mean that neurons are both pre and postsynaptic, just depending on what stage they're in?
Yeah, exactly. Yeah, most neurons in the central nervous system, they're both, depending which end you're on, presynaptic, and then I'm up. All right, exactly.
Gotcha. Thank you.
Yep.
Are all of the neurons in, like, cats, for example, myelinated, or are there non-myelinated ones as well?
Cats are a mystery, aren't they? No, all of the they're exactly like yours and mine. The ones in their brain, assuming a cat has a brain, the ones in the brain are, there's portions of the brain. If I'm going to, let's see, draw a brain. Delete. Clear. So if you think about a brain, here's your brain, and so all of the, if you do a cross section of a brain, and you're looking at the brain in cross section, the neurons out here on the outer portion are generally unmyelinated in any vertebrate, because they're doing a lot of interacting, and the distances are so short you don't need myelin. All it would do is take up space that you need for more neurons. And so that's all unmyelinated stuff. But when you get deep in here, when these, say, neurons from that part of the cortex over here need to talk to neurons over here, for those kinds of connections, you'll have myelinated fibers between. So they typically call this the gray matter, unmyelinated, versus the white matter of the brain, myelinated inside. And that's the same for nearly every vertebrate.
Is there a disadvantage to myelinating cells?